Hello student, I am Dr. Manjusha Vijay Ingole, Head Department of Botany, Kisanveer Mahavidyalaya, Vai. In today's presentation, we will turning towards the BSc part 2nd semester 3rd paper 6. Title of paper is Genetics and Molecular Biology. In this paper, we have to study the first point, chromosome. Chromosome, all of you know that it is present in each cell. The DNA molecule is packed into the thread-like structure called chromosome. Each chromosome is made up of DNA, and that DNA is tightly coiled many times around a special type of a protein called histone protein, and that histone protein support its structure. They appear as a rod-shaped dark stain bodies during the metaphase stage of a mitosis. When these cells are stained with a suitable basic dye and viewed under light microscope, we observe that the thread-like structure present and inside the nucleus, these are nothing but a chromosome. These chromosomes were first described by scientist Strasburger in 1815. And it is a term as a chromosome, and these are firstly used by scientist Waldier in 1888. When we observe these chromosome having the specific shape, size, structure which can be easily understand. Here the chromosome, single chromosome, these are the two arms of the chromosome, and on that two arms we observe these dark as well as a light color bands. Then these are two arms, one arm is termed as a short arm and long arm. These are two arms, short arm is labeled as a small p letter, while the long arm is labeled with a small q letter. These are two arms, these are joined at a point, that point is termed as a centromere. But when the longitudinal splitting of that same chromosome is takes place, then it is termed as a duplication form of that chromosome, and that duplication, it results into longitudinal splitting of that arm of a chromosome, and these centromeres means the point at which these two arms these are joined together, these are remain unsplitted one, and this is the single chromosome, but their duplication is takes place, and therefore it results into forms of two sister chromatids. Then this structure of a chromosome, when we observe this chromosome under electron microscope, we found that the structure, that is a rod-shaped structure, is present. Then this rod-shaped structure is having the outer layer. That outer layer as well as a membrane bond is termed as a pellicle. Inside that, the matrix is present, and in a matrix, the chromatids, thin thread-like structures, these are present, and these are spirally coiled. It is termed as a chromatids. Means in eukaryotes, the chromosomes, these are multiple large. These are linear and are present in the nucleus of the cell. Each chromosome is typically has one centromere and one or two arms that project from the centromeres. Structurally, each chromosome is differentiated into three parts. Outer one is a pellicle, then a matrix, and third chromonomata. The pellicle is nothing but a membrane, outer membrane. It is made up of a non-chromatin material. Inside that, the matrix region is present, and in that matrix, this spirally called chromonomata is present. Chromonomata, here we observe the two threads like structures. These are two threads, these are identical. These are tightly bound. These are spirally coiled, and inside that the 8 microfibrils these are present. Then here we observe that the centromere means due to presence of centromere, the chromosome is having the two parts as well as the two arms. One arm is a longer one arm is a short one. But this is nothing but a term as a centromere. Centromere is also termed as a primary constriction. It is also termed as a kinetochore. When we observe that these chromonemata, when it is passes through the arms of a chromosome, it is a thin thread spirally coiled structures. These are tightly bound. But when the same chromonemata pass through that centromere primary constriction, we found that it is a straight thread-like structures. These are present in these centromere as well as a primary constriction. Means the structure of a chromosome, it is made up of a outer membrane pellicle. Inside that matrix region is present. In that matrix, there is a chromonemata is present. When we observe in the same chromosome, another second constriction is present, and that constriction is termed as a secondary constriction. That constriction is termed as a secondary constriction. Means the nucleolus is always associated with that secondary constriction, and towards the end of one arm of a chromosome, we observe the globular structure that is termed as a satellite. When this globular structure is present, that chromosome is termed as a SAT chromosome. That chromosome is termed as a SAT chromosome. All chromosomes which is present in the cell, these are not having satellite, or these are not SAT chromosome, but only chromosome having the specific globular structure which is termed as a satellite as well as a SAT structure. Then we found that the classification of that chromosome, these are takes place, or types of chromosomes, these are takes place, that is which type of genes present on that chromosome. Then second classification on the basis of a centromere, that is the number of centromere present throughout the length of a chromosome as well as the position of that centromere throughout the length of a chromosome. First, we will turning towards the types of chromosomes. In that first, the chromosome which is having the gene, these are responsible for the body character. These chromosomes, these are termed as autosomes. Means all of you know that in case of human beings, two types of chromosomes these are present. One is termed as autosomes and another one is termed as a sex chromosome. Autosomes means these are nothing but genes which are responsible for the body character. These are present on that specific chromosome, and therefore these chromosomes, these are termed as autosomes. While the sex chromosome means on that chromosome, the genes which is present, these are mainly responsible for maleness, femaleness, sex determination, as well as the character of a sex. Therefore, these chromosomes, these are termed as a sex chromosome. Means in case of a human being, 23 pairs of a chromosome, these are present. Out of that, 22 pairs of chromosomes, these are termed as autosomes, and only one pair of a chromosome is a sex chromosome, either XX or XY. These X chromosome is mainly responsible for the femaleness, while Y chromosome is responsible for the maleness. Then on the basis of a number of a centromere, means throughout the length of a chromosome, how many centromeres these are present. Either one centromere is present, it is termed as a monocentric chromosome. When two centromere is present throughout the length of a chromosome, it is termed as a dicentric. When more than two centromere is present throughout the length of chromosome, it is termed as a polycentric chromosome. When beside this, the chromosomes having without the centromere, then it is termed as a acentric type of a chromosome. And when throughout the length of a chromosome, the diffuse type of a centromere is present, then that chromosome is termed as a diffused or non-located chromosome. Then again, on the basis of location of a centromere, where that centromere is present throughout the length of chromosome, on that basis, the chromosomes, these are classified as metacentric, submetacentric, acrocentric, and telocentric. Metacentric means the centromere is present immediately the middle position of that chromosome, and therefore during anaphasic movement, these chromosomes, these are appear as a V-shaped because the centromere is present at the middle position, and due to which the two arms, these are having the equal length, and Therefore, they show the V-shaped appearance during the anaphasic movement. Submetacentric chromosome means the centromere is present just apart from the centromere. Therefore, one arm is long, one arm is short. And these chromosomes during anaphasic movement, they show the L-shaped configuration as well as the structure. Their one arm is long, another arm is short. Third type of chromosome is acrocentric. Towards the end of that chromosome, the centromere is present. Therefore, these chromosome during anaphasic movement shows the J-shaped structure during the anaphasic movement. Therefore, one arm is a very short one and another arm is a long one. And that type of a chromosome is termed as acrocentric, while the centromere, when it is towards the end, and these are chromosome having only one arm, then that chromosome is termed as a telocentric chromosome. During anaphasic movement, these chromosomes appear as a single rod-like as well as eye-shaped structure during the anaphasic movement. Means on the basis of location of centromere, the chromosomes, these are classified as metacentric, having equal length of sister chromatids. Submetacentric, one arm is long, another arm is short. Acrocentric means one arm is so short, another is long one. And telocentric means having only one arm. Then next, function of chromosome. On these chromosomes, these are specific in each and every cell. Therefore, these are becoming one of the important criteria for the identification of that species on the basis of their number present in that cell. Then, these are the great importance in the determination of phylogeny as well as the taxonomy of that species. These are mainly responsible for the storage of a genetic code. Again, inside that, the sex chromosome is present, therefore these are playing important role in sex determination. These chromosomes, these are again playing important role during the cell division, and therefore these chromosome check successful division of cells during the process of mitosis. And last one is nothing but a formation of proteins and storage. These chromosome directs the sequences of proteins formed in our body, also maintain the order of DNA, and these proteins are stored in the coil structure of the chromosome, and these proteins, these are bound the DNA, and therefore it is helpful in the proper package of that DNA molecule. This is all about the structure, function as well as the types of a chromosome. Thank you. Thank you.